including lung disease, but she had red cell defects, so she had anemia that required transfusion. She had liver failure. She had kidney problems. She did have, like I said, interstitial lung disease. She had some neurologic problems. And then over the, with a lot of support, she started to get better, not from anything we did, but just because whatever was going on with her was getting better. And her lung disease resolved, her anemia resolved, her liver failure got better, but even at 2 years out, she continued to have some developmental delays and growth delays. So we had the opportunity to sequence her exome. We sequenced her, we got DNA from her parents, we got DNA from a healthy sibling, we got DNA from the child. And so here's an idea of what we went through, just to walk through. So we sequenced 30 million nucleotides, so sequenced her exome. From there, we found about 18,000 deviations from the normal published genetic sequence. And we use, there are lots of databases that are publicly available that you can do this against, but so we found 18,000, so we had to do something with that. So here's the first step where we made a couple of assumptions. The first assumption was that the variant had to encode a change in the amino acid sequence or of the protein, so change the protein in some fashion. So that's what we mean by a non-synonymous variant. And so we whittled it down to about 7,500 variants at that point. So there were still 7,500 variants or mutations that encoded a change in the protein. We also made the assumption that anytime there was a missing piece of DNA, and the resolution of these is if you're missing 1 or 2 or 3 of these nucleotides, we still found about 500 of these insertions or deletions, missing or extra pieces of DNA. So then the next assumption we made was, okay, so we have approximately 8,000 variants now. So then we had to say, okay, this has to be a rare disease. We don't run across children with this kind of phenotype very often. And so we have to assume that it's a rare disease. So we went to a couple of different databases that are available and said, okay, these variants have to be rare in the general population and so present in less than 2% of the population. And that got us down to about 1,100 variants altogether. So from that 1,100, that's still too many to work with. So then we made the next set of assumptions, and that is that the variants had to change not only the protein structure, but had to change the protein function. Now, there are no good ways of doing this right now, but there are some computer algorithms that will take your information and try to model changes in the protein and predict whether or not the protein's function is going to be affected by this particular variant. And so there are we use 5 different algorithms. The difficulty with these algorithms is they all come up with different answers. And so we made the assumption that at least 4, if not all 5 of these algorithms had to agree that these variants would predict a change in the function of the protein. And so from there we got it down to 300. And then we made the next assumption, and this is a big assumption. I think it's one of the most significant assumptions where we may go wrong with these kinds of things. And we made the assumption that this was a recessive disease. First, since the parents were normal, they had a normal sibling, we made the assumption that the disorder was recessive, though each parent carried 1 copy of a mutation in a gene, that the presence of 1 mutation was silent. We all have 1 mutation in multiple genes. But when you get the two together in the same gene, it leads to disease. And so we made the assumption that this was a recessive disease. That may or may not be correct, because there are dominant diseases like SBC mutations where it only takes one mutation to cause symptoms. And so that's a big limitation of some of the filtering algorithms that we use right now. So once we did that, we came down to 21 genes that had more than one variant within the gene in the child. And then, based on that recessive transmission, then we looked to see, okay, these genes where there's multiple variants, how do they line up with what the parents are carrying? And if so, what can we find? And in this case, we got lucky. And we came down to one gene that had two variants. Each parent had carried one mutation, and the child carried two mutations. And so this is a gene. It's a gene that's involved in the assembly of proteins in the ribosomes called methynyl-acyl ribosomal synthase. We had never heard of this gene before, but fortunately we work with some very smart people who had heard of the gene, and actually at WashU were working with this gene in animal models, and so we think we came up with the gene in this case. So we have the gene, what next? And so the first thing that we have to do is, first thing to take note of is that even in all of this sequencing technology, there are mistakes. And so we had to go back and validate it, so we used a different type of technique, sequencing this child's DNA, but instead of looking at all 30 million nucleotides, we could look at this one very short region of the DNA and confirm that the child had these mutations, confirm that the parents carried these mutations. So a very important piece. The next thing is, okay, we have a name of a gene, what's known about it? And in this particular case, we were lucky because there had been something known about it. It had a known function. There had been an animal model that had been developed. But I can tell you we've had a couple of cases where we've done this, where we've come up with 4 or 5 candidate genes who kind of made it through the algorithm. And then you go to the databases and it goes unknown function. And then you're left kind of holding the bag there. And then it becomes even more uncomfortable to tell the families that, okay, we went through all of this and we still don't have any appropriate answer. So if you get lucky, somebody's already done something with the gene and you can find out about it. And if you're even luckier, then you can find a way to test this gene. So you have a variant that's made its way through all of the algorithms, but then the next most important question is, okay, does this mutation or this pair of mutations do what we think it's going to do? Does it disrupt some type of cellular process in some type of model system? And so that in some cases that might be an animal, in some cases it might be cell culture. So that's the next step. And then finally, you get all of this information and then you take it back and then says, does all of this make sense in terms of what the phenotype of the patient is? So where we are with this particular case, and I have nothing to do with any of this, so this is all lots of people who are much smarter than I doing things in another lab at WashU. They've gotten through the point where they've have a mouse. They've also tested this in cell culture. But now we have all of that information, so the question is, how can we relate it back? And does this make sense in terms of explaining the child's interstitial lung disease? Does it make sense in terms of the anemia, the liver failure, and growth failure, and so on? And I'm not sure that we have the answer to that yet, but that's what we're working on at this point. So then, as Larry alluded to, okay, we have all of this information, and we have the gene, and we have the function, and we know we can explain everything. How are we going to do this? And then this is the big issue. So then, is there a way to take this information and then bring it back to the patient so that then we can develop some kind of treatment for it? And so you start with animal studies, try to demonstrate changes there. You may start with cell culture studies. Once you think you've identified something that might be safe to use in humans, and you have to go through the Food and Drug Administration, quite a process there to then do the safety trials in humans, and then you have to do the larger scale human trials. And so a lot of work that kind of goes from there. So getting the information, so the genetic technology is getting us to the point where we're going to have lots of information. It is going to provide, it has already, and will continue to provide breakthroughs. I think as the informatics become more and more sophisticated, we'll be able to develop better filtering or better algorithms to really understand these. But it also takes the interaction between computer people, informatics people, biologists who understand the basic biology of the disease, working together so that then you can really refine some of these algorithms and really take the information, get the biology together, and get it back to, ultimately relate it back to the patient and into some kind of treatment. And I think also what this has highlighted is that, so we have this technology available, and we're getting to the point of personalized medicine. And so no longer are we going to, or is it going to be, I go to see my doctor, and we have our few doctors, and we have our relationship and develop the treatment. We're going to have teams of people that are going to have to interact for each particular patient. There's going to be the frontline clinician, so the pulmonologist, the general pediatrician, the neonatologist. There's going to have to be a geneticist who's really understanding what these implications are. They're going to have to be basic scientists, physiologists, cell biologists, molecular biologists, who can put this in a context for a particular patient. They're going to have to be informatics, a lot of computer people. So they're going to have to be large teams of multidisciplinary expertise to be able to interact with an individual patient and really identify what the problem is, and then take it to that next step. So we wanted to leave a little bit of time for questions, just to give you a sense of where we are and where we're going, and hope that someday for everybody here who has unknown lung disease that we'll be able to tell you what that might be.
The computer algorithms that you were talking about, 5 different algorithms, what were those algorithms meant to do, designed for? As far as I guess what I'm thinking is geographically informations of statistics based on the genotypes of certain, is that what it's?
No, what they do is they take a lot of different information together. So the first thing they'll, well, I don't want to say the first, because there are several things that they do. One is there are also assumptions built into those. So one is that if there's going to be a functional change, it's going to be rare. And so they're looking at whether this particular amino acid or part of the protein has been conserved through evolution. So if you see that, going back to lungfish, this amino acid in this protein is present in lungfish, and it replicates all the way above the evolutionary scale, then it gets a certain score. Secondly, it looks at the charge of the amino acid and then tries to predict, is it going to change the folding of the protein, and several other things.
One of the problems is they're not perfect. I mean, we know of individuals who have changes that clearly seem to be causing illness in those individuals, and the computer programs say, oh, they're okay. And there are other ones where the computer programs say this is a problem, but we know individuals who have them and they're fine. But it is one of the issues actually where all the information that you're all contributing helps. Because what we do is we deposit this in public data banks, and those are part of the resources that those algorithms use to help trying to decide whether something is important or not. So you're all contributing to this every time you have genetic testing. and that information gets shared with the greater community at large.
Okay, so this was part of my question, is about sharing this information. Several of our kids, and including my son, have been approved to do, and we've done the whole whole exome sequencing studies. Problem is, our geneticist is not tied to the child network. Of course, I'm going to have him talk to Dr. VC, and I imagine it's the same case with other families. So we've connected through Facebook, and we said, oh, hey, my child had this done. We're waiting for the results, and we're going to compare the answers, but we're not scientists or geneticists. We're probably going to be looking at each other and going, oh, I don't know. So my question is, how do we get this information to people who can do something with it, like you guys, so we can compare and maybe come up with something?
The centers that are doing this through public funding are uploading these into databases in an anonymized way so as to protect privacy, and that is a huge issue, so that the information is available to all the research community. And I mean, the NIH feels very strongly about that, and most of us as researchers feel strongly about that. One of the problems is that the technology has become so available that there are people who are doing it a little bit more in isolation, and the only thing we can do is to try and encourage sort of sharing of the information and sharing of resources. And we're certainly welcome to help out and advise, because that's the only way we're going to do this, is look at it that we're all in it together.
Yeah, and that's where also working with, as I mentioned, it's going to have to be a team of people. So Tim's going to get the information. I'm sure he'll figure it all out, so you won't have to go any further than that. But on the rare opportunity that it might not be completely clear, he does have access to other people. But again, that's one of the things that's the benefit of an organization like this, that you do have the access to it. There are a lot of times this is being done without that kind of background or context.
So knowing with research, we say we identify genes. We've identified the gene that is the culprit, even if there's one, potentially two, like in our daughter's case, 2 mutations on the gene. Is research has advanced enough, and is it moving in the direction of, okay, so we know the gene, we know the mutation that's causing it, even if there's both mutations causing it, we looking into, say, swapping out genes, and has research moving in that direction, saying on animal models of gene swapping to correct that gene, not just necessarily gene therapy, but gene swapping, and trying to, if you will, trick the body into saying, okay, this is what we need to, whether it be on a protein basis or not, to correct that gene, and then trying to solve the problem from there.
So there are a lot of different answers and levels to that question. So I think the first thing is, as I mentioned, really testing that mutation, specific mutation, to see what it's doing to the cell or doing to the animal, and then, is there something from that then we can use some kind of a therapy to try to interact and improve or circumvent the problem. In terms of kind of swapping out genes, there are or correcting mutations, there are technologies that are in progress, and Dr. Cotton is working on some of these right now, where you can cut out the mutation and replace it with the normal sequence. There are actually some medications that are being used for cystic fibrosis, where for particular kinds of mutations, they can basically try to skip over the mutation. So there are things moving in that direction. These are complex issues. And as we've learned the hard way, we've kind of gone into a lot of different treatments with the best intentions, but find that there might be some off-target effects down the road. And so, especially when you start trying to mess with genes in the cells, it becomes a concern that how do you direct it right to that particular cell type in that particular organ without hitting someplace else that's going to have
And I'll go back to what Dr. Haygood showed from the conference about the cloud. Okay, because what we're learning is that you have a problem in the cell where one pathway is blocked. So one of the hopes of this is, can we understand more? And this is where Dr. Cotton's research, because actually being able to get cells that have the particular defect in it and let us know, then how can we take advantage? How can we find those other pathways down the hill? So even if this one isn't working, what can we do to sort of redirect the cells to a state that's going to be more functional? And I think actually my own personal view is that's actually going to be a more fruitful approach for a lot of these illnesses than trying to sort of replace the specific. But that's where the informatics comes in too, because as Aaron said, we're looking at single genes. But we know individuals can have the same genetic defect. We have situations where we have identical twins. Yet the outcomes, the clinical manifestation of illness is very different. So there are other things that are factoring in there. So understanding some of the other genes, some of the other pathways is going to be critically important. Nothing is working in isolation.
I'd like to thank Dr. Hamvas and Dr. Nogi for this great presentation. Everybody else, if you have any other questions for them, they're going to be around for a little bit longer.